I'm going to end the day with something that really doesn't have anything to do with getting old and age-related anything. But it sort of does, because I'm going to talk a lot about development. So I can argue that you begin to age the minute that you're born. It's just that different processes take over throughout the course of a lifetime. And so, as Cam mentioned, I'm an evolutionary neurobiologist, and I'm interested in how brains get complex. How we, 200 million years ago, mammals were very, very small and had only a few parts to their neocortex. And how you get a brain that has hundreds of parts like humans that are interconnected in very specific ways that generates the complex behaviors that we associate with humans. That's my ultimate question, but I'm going to talk a lot about development today. So I'm going to start with a couple of questions. First is, what makes individuals different? If I were born in another country that had a very, very different culture, would I be the same Leah Krubitzer? If I were born in a different time, would I be the same Leah Krubitzer? And I'm going to attempt to answer some of those questions today, and they might not be the right answers, but I think we're sort of onto something. So the first question, first I want to point out that we study the neocortex. This is a brain of a bottlenose dolphin, the back of a brain of a bottlenose dolphin, and that is a brain of a very small Australian marsupial called a native cat. And we study the neocortex, which is this outer portion of the brain, and I don't know if you guys are familiar with the neocortex, but it's that portion of the brain that's involved in higher cognitive processes, perception, language, and a lot of the very complex behaviors that we associate with humans. And one of the hallmarks of human evolution is the relative increase in the size of the neocortex relative to our body, or with respect to our body. It's the portion that I said it's changed the most throughout the course of evolution. And it's not just that brains get larger, because lots of animals have big bodies and have big brains, but the neocortex is divided into a number of different parts. And as I said, animals that are really simple, like mice, for example, maybe have about 15 neocortical areas. And humans, the running number is about 300 cortical fields. So how does this process occur over the course of 200 million years? And ultimately, how does it generate the behaviors we associate with the human condition? So that's the question. And I'm going to show you these schematic cartoons throughout the talk. Blue, the different colors just represent different cortical fields. And I'm just showing that you go from a very simple form that was very small to something that's very large and complicated. And it's not just, and this has been alluded to all day, it's not just the increase in the number of cortical fields, it's how they're interconnected. And in fact, people who study our closest living relatives, the great apes, suggest that it's not just the difference in brain size that makes humans different than great apes, like chimpanzees, but it's how these brain areas are interconnected. Very, very important. Okay, so what's the problem with studying evolution? The problem with studying evolution is that it doesn't happen over the course of minutes, and it doesn't even happen over the course of a lifetime. It happens over tens of millions of years, hundreds of thousands of years. So how do I figure that out? Because there's this process that occurs over a very long period of time. There are two ways we can approach this problem. One is what's called the comparative approach. So I can study what evolution has produced. And what evolution has produced is brains and bodies. So I can study tons of different brains and bodies of a lot of different animals. And I can say what features are similar and what features are different. So in terms of similar features, I'm going to talk a little bit about the types of techniques we use. These sorts of comparisons allow us to make inferences about an unknown form, like the common ancestor. Because you might be thinking to yourself, well, how does Leah know what the neocortex of the common ancestor looked like 200 million years ago? Because brains don't fossilize. You can tell me something about the size, but not about what it looked like. A comparative analysis is something like this. So I'm looking at you, and let's say you have 4 siblings. And I never saw your parents, but I can say, okay, well, I'm looking at you, and you're a certain height, a certain stature, a certain body build, a certain eye color. I can make inferences about your mother and father. And then I can look at your cousins and look at similar characteristics and probably make good inferences about your grandparents, and so on and so forth. We can do the same thing with brains. We can look at a variety of different brains, we can see which features are similar, and we can say it is likely that that was evolved from a common ancestor. I'm giving you a little lesson in evolutionary neurobiology. So if I look at 100 species and I say they all have cortical area A, B, C, and D, all of them have it. There are 2 possibilities. One is it was inherited from a common ancestor, pretty simple, no transformations, or miraculously, they evolved it independently 100 times. That scenario is highly unlikely. And that's the basics of how we do comparative analysis and appreciate the unknown form. And before modern imaging techniques, like fMRI and PET and a lot of the things that Tarn talked about, that's how we knew about human brains. almost everything we know about the human brain comes from work on other animals. Because of this comparative approach, because things are inherited from a common ancestor, we can learn a lot about species, humans, by not studying humans directly. But here's the problem with the comparative approach. If I'm looking at an individual animal, that evolution is a moving picture of life. I'm taking one frozen frame and looking at that frozen frame, and I'm looking at a lot of different animals, and I'm trying to put together this puzzle of how evolution occurred. So we're sort of studying frozen moments or frozen frames. Comparative approach can tell us what sorts of changes evolution has produced to brains, but not how those changes occurred. And that's where studies of development come in. The evolution of the neocortex is the evolution of developmental mechanisms that give rise to some aspect of the cortical phenotype. Studies of development tell us how changes have arisen. Presumably, in the development of different species, something's being tweaked during development that allows a species to have lots of cortical fields, or the sorts of brain changes I'm going to talk about in a little bit. So that's kind of what I'm going to focus my talk on right now. So what am I comparing? I'm comparing functional organization using electrophysiology. So a lot of these animals, we expose their neocortex, we stick an electrode in the brain, we crank up the amplitude about 10,000 times so we can hear neurons, we can see what they want to respond to, and we pepper the cortex with hundreds of recording sites. And we say, okay, which portion of the brain are neurons responding to visual stimulation versus tactile stimulation versus auditory stimulation? That's just very grossly what we do. So we make maps of the neocortex. I can make there are maps of the body, there are maps of the visual field, as Greg mentioned, there are maps of the cochlea in the cortex, and that's how we do that. I'm comparing cortical connections. So if I have hundreds of cortical fields, I can actually inject substances into the brain that tells me what regions of the brain are connecting to that particular region of interest and what it's sending projections to, so how it's interconnected. And looking at cortical architecture. If I stain the brains in a variety of ways, Gray showed you some sort of architecture, and I think others did as well, brains look different. You don't have to be a neuroanatomist to actually see with special stains that some regions are dark, some regions are light, some regions have different cell structures, some regions utilize different neurotransmitters and so forth, okay? So that's what I'm comparing across the species. And so, God, I guess it's about 30 years now of comparative work. My mother's so proud of me, this is all I produced. Not $1 million, not a nice house, and I do not drive a fancy car, but I have my Leah Krubitzer cladogram. I'm loving it. So you don't have to know what all these fields are, but the cortical fields that are homologous are marked in blue. And what you can see is that there's a common plan or constellation of cortical fields that all species have, likely inherited from our common ancestor. And before we knew about the human condition, we proposed that humans had it as well. And now we've subsequently tested that they do in fact have it. What's really important and cool, and this is just a little aside, is that even in the absence of use, so if I'm a blind mole rat and my eyes have gotten really small and skin has grown over my eyes and I don't use my visual system, I still have a visual cortex. I still have certain pathways associated with visual processing. They've been taken over by the auditory system, but I still have them. I can't get rid of them, probably because of genetic constraints. Okay, but species are different. So this is a macaque monkey brain that's been, you take the cortex, we peel off the brainstem and the thalamus, we flatten it and we cut it parallel to the cortical surface. This is a little mouse brain not gone to scale. And the reason we do this is that it allows us to appreciate spatial relationships of fields. So if I took a globe, I could slice it up and I could say, okay, well, where is Africa relative to North America? Where is North America relative to South America? It's kind of hard to do. So cartographers have been doing this for a long time. They take the map, they peel it off the globe, they make a couple cuts and they flatten it. So that's what we've done. So this is the back of the brain, this is the front of the brain, the top of the brain or medial is this way and lateral. So this is a macaque monkey, and the first thing you're going to say is, wow, there are a lot of differences. Sure, I can see these common cortical fields are homologous, which means inherited from a common ancestor, but there are a number of different changes to the neocortex at a systems level. And so schematically, I've represented them here. My husband was at my last public talk I gave, which was actually at a bar. It went pretty well. You guys were all drinking. You guys were drinking right now. You guys got to be half in the bag to really get this. And he said, this is too complicated. Don't show him this. This is not complicated. The squares. Let me tell you why. The squares are meant to represent a cortical sheet. And so some of the changes that have been made to the cortex, I can use my pointer, are changing the size. So here's a large cortex and here's a small cortex. I already talked about that. Changes in sensory domain allocation. Don't let that make you crazy. Red is somatosensory, yellow is auditory, and blue is visual. And what this is showing that this cortex, let's say they have the same size cortical sheet, has a lot of cortex devoted to visual processing, this much devoted to somatosensory, this much to auditory, and this one has a lot of cortex devoted to auditory processing. So change in the allocation of what sorts of sensory inputs you're going to process a lot of. Change in the relative size of cortical fields. If you have the same size cortical sheet, in this animal it's bigger, this blue field is bigger and this one's smaller. Magnification of behaviorally relevant body parts or sensory receptor arrays. So the amount of my brain that's devoted to representing important things that I do is magnified. So for humans, in the somatosensory system, the tongue, the lips, the digitives, they're magnified. In a rat or a mouse, where they're whisker related, they have a huge representation in their somatosensory cortex of the whiskers. And I'll show you more examples of that. And we can see that across sensory systems. I'm not going to talk about this. A number of cortical fields I already talked about and cortical connections. And what I'm going to focus on today is how sensory domain allocation, magnification of behaviorally relevant body parts, and connections occur. And so, what factors contribute to these modifications? Is it all genetically hardwired through inheritance? Genes intrinsic to the neocortex already have a plan of action, doesn't matter what happens, they're going to unravel in a particular way, and you're going to have these sorts of cortical fields with a certain sort of connectivity that look a certain way. Genes extrinsic to the cortex. In my first slide, I showed a lot of bodies, a lot of animal bodies, because the brain is enslaved by the body. We can't talk about the brain at all unless we consider that it is part of a body, that's part of a social structure, that's part of the planet Earth. I mean, we can isolate it to try to study it, but we have to look at it in the context of the body. And then I'm going to focus mostly on epigenetic alterations or activity-dependent changes. Are there things that occur during development that can change cortical sheets, the cortical field size, cortical connectivity, sensory domain allocation? A lot of the things that we see across species that we often associate with traditional evolutionary sort of inheritance as opposed to activity dependent. So what we do in our laboratory is we try to play out evolution. We induce modifications in the developing nervous systems. We're making those tweaks that we think are being made naturally throughout the course of evolution, and seeing if we can generate a cortex that isn't just viable, but that looks like something evolution would produce. Can we change the size of cortical fields? Can we change connectivity? Ultimately, can we make new cortical fields? And then, obviously, we'd like to look at how that impacts behavior. Okay, so here's my talk really quickly. I'm going to give you some examples from the natural world. I'm going to show you some experimental manipulations, and I'm going to show you studies about natural variation in early sensory experience within a population. Okay, changes. This is my favorite example on the planet Earth, and it's a duck-billed platypus. Cam's from Australia. So I went to Australia to study the duck-billed platypus. No one except me understood why. And I think when I really began to appreciate that bodies were important was that I had to catch all the animals I worked on. And in fact, another thing my mother is quite proud of is that I have been peed on, pooped on, and puked on by more animal species or mammalian species than anyone on this planet. Crocodile guy had me beat, but he bit the dust. So that is also my claim to fame. Cam said, oh, she's famous. Now you know why. So let me tell you something about the platypus. It's a really interesting creature. So it has a bill. This is a real platypus. And it has stripes of receptors on its bill running like this. And they're really responsive to touch. And they're interdigitated with electrosensory receptors. They can passively detect small currents in the water to pick up for prey and other sorts of things. When they're doing anything important in their platypus life, they close their eyes, so no visual input, their ears, no auditory input, and their nose, no olfactory input. And they go like this. So they're living by their bill. And you say, what does that cortex look like? Here's what it looks like. If I stuck a ton of electrodes in the brain, and I mapped out their body, this is a little representation of their body. You can see their tail up there, their paws. And if I look at their bill, this is the front of the brain, this is the flattened cortex, this is the top of the brain. In just this one field alone, I have this enormous representation of the bill. It takes up 90% of the somatosensory cortex, or primary somatosensory area. I have another huge representation of the bill, another representation of the bill here. So about 70% of the entire cortical sheet is devoted to processing inputs from the bill. This is what I mean by behaviorally relevant, or magnification of behaviorally relevant body parts. And we study these sort of specialized animals to give us some insight into more subtle changes that might be occurring throughout development and throughout the course of evolution. So again, the question is, to what extent is that enormous bill representation associated with genes intrinsic to the neocortex that are just saying you're S1, you're the bill, you're primary visual cortex, genes extrinsic to the cortex, but genes associated with the development of the body. I mean, you have to have genes that are making that bill. Epigenetic influences on the body. Sensory stimuli present throughout development and peripheral morphology. And I should say, anytime you have a specialized body part, you use it in a very specialized way. So I have specializations of my supralaryngeal tract, my oral structures. They don't just sit around doing nothing. I use them for speech. So there have been studies in other laboratories. You can muck around with genes. People do it all the time. They have these special mice that are transgenic mice, and they can overexpress genes associated with cortical development, underexpress them, do all kinds of things. And the bottom line is, yes, of course, genes play a role in some of these factors of cortical organization that I told you about. But interestingly, if you look at comparative studies of forelimb morphology in a rat and or a mouse and a bat, you can say, okay, here's a forelimb of a mouse, big little paw thing, and then you have this exquisite wing of a bat that looks really quite different, really phenotypically different. Elongation of the digits, the growth of interdigit membranes. You have specialized little touch domes that are exquisitely sensitive to touch on the wing, so that if you're flying, you can make wing adjustments very rapidly. And then what they did is they looked at the expression of genes associated with forelimb development. It turns out there are slight tweaks in where those genes are expressed, the extent of the limb they're expressed in, and there are about 3 or 4 of them that are tweaked that can turn this into this. And so you're saying, okay, so that's interesting. And if you look at the cortex, and you look at the forelimb representation, it has a much larger forelimb representation than a mouse. Is that due to genes intrinsic to the neocortex? Or is it simply changes in the body and use that generates this sort of thing? And of course, they have a large portion of cortex devoted to the auditory system. Okay, so I gave you a basic premise. I'm going to be done in soon. Okay, I started a little late though, right? Yeah. Okay. This is the point I want to make. I gave you an example of the bill of the platypus. We've seen these sorts of magnifications, these behaviorally relevant magnifications. I think there was a poster from deep out in the lab on this. In a star-nosed mole, where you have this nose that does this specialized tactile reception, and you have a huge representation. The point I want to make here is humans also have their specializations associated with speech. And when we think about human specializations, we can't think that we've stepped out of evolution, that we have somehow, we're not like other animals, we don't evolve and are enslaved by the same principles. We are. We can look at Broca's area, classically defined Broca's area, and say, well, it's just following the same rules of construction as the bill of the platypus. Is an expansion of S1, M1, premotor cortex, changes in connectivity, but it is in fact following those same rules. So now I wanted to show you just a little bit about experimental manipulations that we do in our laboratory. These are bilateral nucleated animals. This is a short-tailed opossum. The reason we work on them is because they're born like this. They're born as little embryos. Their nervous system is not connected up at all. And here's what we do. We say, okay, I want to get at that question, to what extent are those, magnification of behaviorally relevant body parts, cortical connectivity, sensory domain allocation, due to changes in activity that the brain has access to early in development. So here's what I'm going to do. I'm going to remove all visual input really early in development before any of the connections are formed. I'm going to let the animal grow up and I'm going to see what happens. So that's what we do. And we measure cortical field size. This is just Latin cortex, and you can see V1, V2, these other things. And this is a normal animal. This is a bilaterally nucleated animal. I can see, okay, this field's, what should have been visual cortex is much smaller. What is auditory cortex with somatosensory cortex is much bigger. The size of the cortical sheet hasn't changed, but I've changed the size of cortical fields. But what's even more remarkable is when we stick an electrode in the brain and we record from all the neurons in what would normally be visual cortex, which is in blue here, all of this cortex has been taken over by the somatosensory and the auditory system. So I've done nothing to the cortex itself. I've simply changed the ratio of sensory inputs coming into the developing cortex. And what we find is if we look at what this region of cortex is representing, it's a big, huge face representation with rubricity. So this animal, we've taken an animal that's highly visual, and we've changed it into a neocortex that's all about the face, the rubricity, and the hairs. And if we put anatomical tracers in what would be this primary visual cortex, these are normal connections on this side, and these are abnormal connections. And what we see is that this field is receiving inputs from its normal targets, But it's also receiving inputs from the auditory system, the somatosensory system, at both the cortical and subcortical level. I totally rewire the system. And what's really cool is recently in our laboratory, we are recording from neurons in S1 and looking at connections of S1. They're altered as well. So you haven't just altered the visual system, you've also altered the somatosensory system. So it's very important because blindness is not just the absence of light. So when you say, what's it like to be blind? I'm just going to close my eyes and I'm blind. Well, you're not. Because your entire system has been rewired due to this loss of input. What we're looking at now, so the idea of retinal prosthetics or gene target therapy for the eye are fine, but if you were born blind, it's not really going to help with this remodeling. What our laboratory is working on now is to see if we can take this natural plasticity and harness it, direct it and amplify it so we can make you a super tactile blind person or a super auditory blind person so you can more readily generate adaptive behaviors with the remaining nervous system. And in the last 2 minutes, I'm just going to quickly talk about something that's a little bit more refined. And these are prairie voles. And I'll tell you something about prairie voles. This is like, if you want a game show and you're listening to this talk, take animals for 100 because you're going to get it. Let me tell you about prairie voles. So prairie voles there are 3% of mammals that are monogamous, and prairie voles are one of them. They're also biparental, so the mother and father both rear the young. What's cool about prairie voles is that you can measure the amount of contact that prairie voles have, and like anything in biology, it follows under a normal distribution. There are some parents that are high contact, meaning you can measure how often they're nursing, how often they're grooming, how often they're huddling, and there's some that are low contact. And you can't put a positive or a negative value on that because in evolutionary biology, nothing is good or bad, it just is. But you can measure that. And so of course you want to say, okay, what does the somatosensory cortex look like? But let me just tell you something cool about this contact. If you take known high contact babies, babies from known high contact parents, and you put them with low contact parents, and if you take low contact babies and put them with high contact parents, they become the type of parent that they were reared with. So I missed a step there. If you're a low contact baby, you become a low contact parent. If you're a high contact baby, you become a high contact parent. So if you cross foster them, you become what your foster parents are, not what your biological parents are, suggesting that this behavior is culturally transmitted. So we got this, so we already know what we're going to be looking for because we took the sledgehammer to the system by removing eyes. So what we do is we record from neurons in the brain, and we also look at connectivity, and this is basically just showing you, and this is a paper we recently published, that if you look at the high versus low, especially in this region down here, this is a way of just quantifying connections. We injected the primary somatosensory area, and what we see are 80% of the connections look similar in both of these groups, but we see many, many more connections with entorhinal cortex. So presumably these connections are culturally transmitted, and in conjunction with a woman in psychology, Danielle Stolzenberg, we're looking at the molecular mechanisms that underpin these differences in connectivity. So what factors contribute to the cortical phenotype? Like I said, genes can contribute to these sorts of cortical field size, connections, and those sorts of things. But the environment can contribute equally to those sorts of things. And I've given you examples of the bill of the platypus, the tongue and larynx, and lips of a human. But what about social learning, language, and culture? I would contend that in order to really understand how these impact the brain, we have to think of these as simply complex patterns of physical stimuli that are impinging on our developing nervous systems. So love is great, but I would rather think of love as temperature, cadence of a voice, visual stimulus, tactile stimulus that are impinging in a combination on a developing nervous system and changing how your brain is interconnected, changing size of cortical fields, probably changing neural response properties. And one of my favorite examples of why I think this is so is this. This is a human cladogram, and this is what's happening. Hominins here about over a million years ago to the present. On the top is genetic, anatomical things that have been going on, and at the bottom are cultural things that have been going on. And what's really remarkable, if you look about 3.2 million years ago, we had features of the modern hand. Only about 1 million years ago, we had control of fire. We've had the modern hand in the modern vocal system for 700,000 years. It's not clear. They say modern language may have evolved around that time, but more people are putting it here. The point is we've had these anatomical things and genetic things in place, but it wasn't until about 200,000 years ago where things really started ramping up. So we have been humans, genetically defined humans, for 200,000 years. No real huge changes up here, but tons of changes down here. So you're getting, even 300,000 years ago, we were still using spears, right? Industrial revolution has only occurred about 200,000 years ago. So if you believe, if you believe that all behavior is generated by the brain, and this behavior here, in the present, is radically different than this behavior here, there must be some sort of brain changes here due to cultural evolution that aren't changes in gene sequence or evolution, but simply the propagation of those brain phenotypes that are built in development over successive generations, that allows us to do these sorts of things. Okay, so this is my laboratory. I'm done with my talk.